Biochemical methods are the laboratory techniques used in biochemistry to study the structure, composition, function, and chemical processes of living organisms. These methods help scientists analyze proteins, carbohydrates, lipids, enzymes, DNA, RNA, hormones, and other biological molecules. Here are the major biomedical methods explained in detail. Centrifugation is a technique used to separate particles from a solution based on their size, shape, and density using rapid spinning. Let's discuss types of centrifugation. First, differential centrifugation separates cell components by size. Larger particles settle first. For example, the nucleus settles first, then mitochondria, then ribosomes. Density gradient centrifugation uses layers of different densities. Particles move until they reach their own density level. It's used for DNA purification and lipoprotein separation. Centrifugation has many applications in science. First, it isolates organelles from cells. Next, it separates blood into plasma and cells. Finally, it purifies proteins for research. It's a powerful tool. Number 2 is chromatography. Chromatography separates substances based on their movement through a stationary phase and a mobile phase. Paper chromatography uses filter paper. It separates amino acids and sugars. Let's discuss thin layer chromatography, or TLC. It uses a silica gel plate, making it faster and more sensitive. Next is column chromatography, which uses a glass column packed with adsorbent. Finally, gel filtration chromatography separates molecules according to size. Larger molecules elute first, while smaller ones take longer. Next, affinity chromatography separates proteins based on specific binding. This technique is highly selective. High-performance liquid chromatography is a precise and fast technique. It's used in pharmaceutical and clinical labs for applications like drug analysis, protein purification, and amino acid separation. We'll explore how it works. Number 3 is electrophoresis. Electrophoresis separates charged molecules using electricity. Let's look at the diagram. One type is agarose gel electrophoresis, used for DNA and RNA separation. Let's learn about SDS page. It separates proteins by their size. The principle: charged molecules move toward opposite electrodes. It's used for DNA fingerprinting, protein analysis. We have also protein analysis and genetic studies. Then we go into number 4, biochemical methods known as spectrophotometry. This method measures how much light a substance absorbs. Different substances absorb light at different wavelengths. Its applications is in measuring glucose concentration, enzyme activity analysis, protein estimation, Beer-Lambert law.
The Beer-Lambert law states that absorbance equals the product of molar absorptivity, concentration, and path length. To demonstrate, we generate a light length increases, the light is absorbed more, increasing the overall absorbance.
Colorimetry measures the concentration of colored substances. The principle is that color intensity is proportional to concentration. Observe these beakers. The darker the yellow, the higher the concentration. This demonstrates the colorimetry principle. Colorimetry has many applications in medicine, like blood glucose testing, hemoglobin estimation, and cholesterol testing. The difference from spectrophometry is that a colorimeter uses visible light only, while a spectrophometer uses both UV and visible light. Number 6 is pH measurement. It determines acidity or alkalinity. The instrument is a pH meter. Applications include buffer preparation, enzyme studies, and clinical diagnosis. The pH formula is pH as native log as log. Number 7 is enzyme assays. Enzyme assays measure enzyme activity. Types: A. Continuous assay, reaction monitored continuously. B. Discontinuous assay. Enzyme assays are vital tools in science. They are used in diagnosing diseases, drug testing, and metabolic studies. For example, liver enzyme tests help detect liver damage, and the amylase activity test can diagnose pancreatitis.
Number 8 is radioisotope techniques. Uses radioactive substances to study biochemical reactions. Applications include hormone studies, DNA replication studies, and cancer research. Number 9 is immunological methods. These techniques use antigen-antibody reactions, immunosorbent assay. It's used to detect conditions like HIV, hepatitis, and hormone levels. This reaction provides accurate results. Then we have Western blotting, which detects specific proteins. C. Immunofluorescence, which uses fluorescent antibodies. Number 10 of biochemical methods used to study genes and nucleology techniques. PCR, or polymerase chain reaction, amplifies DNA. DNA sequencing determines the order of DNA bases. Recombinant DNA technology combines DNA from different organisms.
Let's explore some key applications of genetic analysis. These include genetic diagnosis, forensic science, and biotechnology. Number 11 is microscopy techniques. Microscopy helps visualize cells and molecules. There are 2 main types of microscopy. Light microscopy uses visible light. Electron microscopy uses electron beams for higher magnification. Applications include cell structure studies and microorganism identification.
Finally, we have dialysis and ultrafiltration. These methods separate molecules using membranes. Dialysis separates small molecules, uses pressure to separate substances. Biochemical methods have key applications in kidney dialysis and protein purification. Biochemical methods are essentials. They enable the early detection and treatment of various conditions.
They also assist in drug development, aid medical research, improve biotechnology, help understand metabolism and genetics. Conclusion: biochemical methods are powerful laboratory techniques used to study life at the molecular level. They help scientists analyze biological substances, diagnose diseases, understand metabolic pathways, and develop new medical treatments. From centrifugation and chromatography to PCR and immunological tests, these methods form the foundation of modern biochemistry and medical science. Thank you for listening.